Hi, my name is Celeste Suart. I'm a PhD student from the Truant Lab at McMaster University in Canada. Today I'll be talking with you about how ataxin 1 is signaled to DNA damage by ATM kinase. Ataxin 1 is the affected protein in spinocerebellar ataxia type 1. The inspiration from this work came from findings from Betancourt and colleagues showing that SNPs and DNA damage repair proteins could modify the age onset of symptoms for SCAs. As the Truant Lab had previously characterized the DNA damage response of the Huntington protein in Huntington's disease, we wanted to apply this skill set to the SCA1 context. I only have time to cover a few highlights of the data today. However, the full manuscript is available on bioRxiv if you're interested in learning more. First, we asked if ataxin 1 has any particular response to DNA damage events. To do this, we used two different assays. The first technique is the microradiation assay. This is where we use a 405 nanometer laser to irradiate a portion of a nucleus in cells, causing localized DNA damage. We then measure fluorescence intensity for EGFP-tagged ataxin 1 constructs to quantify movement of the protein into the area of damage. We did this for both wild-type ataxin 1 and for disease-associated expanded ataxin 1. Both forms of ataxin 1 were able to localize to damage, as shown here. However, expanded ataxin 1 had less localization to sites of damage compared to wild type. The second technique is the chromatin retention assay, where we measure if a protein is bound to chromatin following an oxidative DNA damage event using potassium bromate. If a protein is involved in DNA damage repair, we expect that more will be bound to chromatin following an oxidative stress event compared to basal control conditions. Again, we used both wild type and expanded ataxin 1. And both were retained in chromatin following damage events. However, similar to the microradiation assay data, expanded ataxin 1 had less retention compared to wild type, as shown here. Now, up until this point, the response of ataxin 1 was mirroring that of the Huntington protein we had previously worked on. Since the Huntington response was dependent on ATM kinase activity, we wanted to see if the same was true for ataxin 1. When we repeated the microradiation assay with ATM kinase inhibitor, we no longer saw ataxin 1 going to sites of damage. This is the first evidence that ATM could be modulating the ataxin 1 response. Our next question was if ATM was directly interacting with the ataxin 1 protein, or if ataxin 1 was simply downstream of one of ATM's many DNA damage repair cascades. Using the kinase prediction software GPS 3.0, we identified a potential ATM phosphorylation site in the ataxin 1 protein at serine 188. Here we have three canonical ATM phosphorylation sites in other proteins, and here we have the conserved sequence in ataxin 1 through multiple species. We hypothesized that serine 188 was the ATM phosphoserine, with serine 186 potentially playing a priming role. We then generated serine to alanine point mutants for both of these sites, and when we did, these ataxin 1 S to A mutants had impaired responses in both the microradiation and chromatin retention assays. With this information, we felt confident in investing in the generation of a phospho-186, phospho-188 ataxin 1 rabbit antibody. We then went on to do multiple experiments with this reagent, including asking what happens to this phosphoepitope under stress conditions if we reduce ATM protein levels through siRNA knockdown. As you can see here with the scramble siRNA, we see an increase in phospho-186, phospho-188 ataxin 1 under conditions of oxidative stress. This response, however, is lowered during ATM siRNA knockdown. We also did this experiment with ATM kinase inhibition instead of siRNA, and we saw similar results. To see if reduction of ATM protein levels had any impact on disease phenotype, we turned to our colleagues at the Botas lab at the Baylor College of Medicine. Using their SCA1 Drosophila model system, they conducted motor climbing assays, where the speed at which flies are able to climb up a glass tube is measured using computer tracking software. They showed that the reduction of the Drosophila ATM homolog TEFU, either by shRNA knockdown or through a genetic cross, was able to significantly improve motor phenotype in the flies. More work is needed to clarify mechanism as to what ataxin 1 is doing at sites of DNA damage and its interactions with ATM. But this work identifies ATM as a potential therapeutic target in SCA1. Thank you so much for listening. I'd be happy to take any questions you have at the coffee break.